വെൽക്കം ടു മൈ ക്ലാസ് റൂം.
ഹലോ കൂട്ടുകാരെ, നമ്മളിന്ന് ക്ലാസ് 8 ബയോളജി ലൈഫ് മിസ്റ്ററീസ് ഇൻ ലിറ്റിൽ ചേമ്പേഴ്സ് എന്ന ചാപ്റ്ററുടെ ഷോർട്ട് നോട്ട്സ് ആണ് ഡിസ്കസ് ചെയ്യുന്നത്. ദ ബോഡി ഓഫ് ഓൾ ഓർഗാനിസംസ് ആർ മെയ്ഡ് അപ്പ് ഓഫ് സെൽസ്. മൈക്രോസ്കോപ്പ് ഈസ് യൂസ്ഡ് ടു ഒബ്സർവ് ദ സെൽസ്. Microscope in which more than one lens is used are called compound microscopes. A compound microscope magnifies an object only 2000 times maximum its size, while electron microscope, which utilize electrons instead of a light source, can magnify objects million times clearly. Parts of a compound microscope: eyepiece, knobs, objective lens, stage and clip, condenser, mirror. Eyepiece and objective lens are the two types of lenses seen in a compound microscope. The lens in the condenser focuses light on the material to be observed. Diaphragm of the condenser helps to regulate the intensity of light. Mirror is with a plain mirror on one side and a concave mirror on the other side. Stages for the preparation of the observation material. For instance, a cross section of plantain root. Put the material in water to prevent dehydration to the material. Take thin sections of the material and keep it in a watch glass containing water. Put the thinnest complete sections to a watch glass containing steam to get color to cells. Add 1 or 2 drops of glycerin to the slide to prevent the drying up. Wash one of the stained sections and place it in the glycerin on the slide. By using a needle, place a cover glass over it without forming air bubbles. By using a tissue paper, wipe out the excess glycerin. Place the slide and observe it through the microscope. Milestones in the history of cell biology. 1665, Robert Hooke observed cells with the help of simple microscope. He called the tiny chambers as cells. 1831, Robert Brown discovered and named the center of the cell nucleus. 1838, M.J. Sheldon find out that the body of plant is made up of cells. 1839, Theodor Schwann find out that an animal body is made up of cells. 1858, Rudolf Virchow observed dividing cells and inferred that new cells arise only from existing cells. The cell theory, formulated by M.J. Sheldon and Theodor Schwann. The body of all organisms is made up of cells. Cells are the structural and functional units of organisms. Functions performed by the cells: resist foreign particles, release energy from nutrients, synthesis biomolecules. The covering of a cell is called cell membrane or plasma membrane. Why plasma membrane is called a selectively permeable membrane? As they allow only the essential materials to pass. Protoplasm, all substances inside the cell membrane, that is cytoplasm plus nucleus equal to protoplasm. The controlling center of a cell is called nucleus. Prokaryotes are organisms without a nucleus in their cell. Example: bacteria, cyanobacteria, mycoplasma. Eukaryotes are organisms with a well-defined nucleus in their cell. Example: amoeba, animals, plants. Parts of a nucleus: nuclear membrane, double layered covering; nuclear pore, pores in the nuclear membrane; nucleoplasm, fluid part of nucleus; nucleolus, spherical bodies that help in ribosome synthesis; chromatin reticulum, gene carrying network in the nucleoplasm; cell organelles, specific parts seen in the cytoplasm. Cell organelles seen in both the plant and animal cells: mitochondrion, endoplasmic reticulum, ribosome and Golgi complex. Cell organelles seen only in plant cells, vacuole and plastids. Cell organelles seen only in animal cells, centrosome and lysosome. Difference between plant cell and animal cell. In plant cell, cell wall is present, no cell wall in animal cell. Vacuole and plastids are present in the plant cell and it is absent in the animal cell. No centrosome and lysosome in case of plant cell. Centrosome and lysosomes are present in animal cell. Plasma membrane, nucleus, mitochondrion, endoplasmic reticulum, ribosome, Golgi complex are seen in plant and animal cell. Different types of cell organelles and their functions. Cell organelle function peculiarity. Mitochondrion, production and storage of energy, powerhouse of the cell, abundantly seen in the cells of liver, brain and muscles. Endoplasmic reticulum, conduction of materials, the passage in the cell, provides firmness and shape to the cell. Cytoskeleton. Ribosome, protein synthesis, seen attached to the endoplasmic reticulum or free in the cytoplasm. Golgi complex, collects cell secretions like enzymes, hormones, mucus, etc. in small vesicles, seen in plenty in glandular cells. Vacuole, stores water, salt, excretory materials, etc. Its membrane is called tonoplast. Plastids, chromoplast, imparts color to flowers and fruits. Chloroplast, performs photosynthesis, gives green color to plant parts. Leucoplast, no specific color, seen abundant in storage cells. Centrosome, contains the centrioles that helps in cell division. Lysosome, contains digestive enzymes that destroy foreign substances entered into the cell. Examples for pigments present in different chromoplast. Xanthophyll, yellow, carotene, yellowish orange, anthocyanin, red, purple. Pigments seen in the chloroplast. Chlorophyll, green. The secret behind the change in the color and taste of fruits. As fruits ripen, chloroplast change to chromoplast to change its green color. Starch in the fruits is converted to sugar to change its taste. Plant cell have cell wall, plasma membrane, protoplasm, cytoplasm, nucleus. In cytoplasm, cell organelles are mitochondrion, endoplasmic reticulum, ribosome, Golgi complex, vacuole, plastids. Animal cell have plasma membrane, protoplasm, cytoplasm, nucleus. In cytoplasm, cell organelles present are mitochondrion, endoplasmic reticulum, ribosome, Golgi complex, centrosome, lysosome.